Thank you so much to the organizers for the invitation to speak and for the kind introduction. Can you hear me okay?
Yes.
Thank you. And please accept my sincerest apologies for not being able to be there in person due to an illness. Today, I will talk about the Mendelian disorders of the epigenetic machinery and share some clinical insights from our epigenetics and chromatin clinic and the discovery of a new syndrome within this group. So every cell in our body contains the same genetic material in the form of DNA. However, cells must do different things at various times in development and in various tissues. Epigenetics plays a key role in this. Epigenetic or chromatin marks are heritable modifications of the DNA or associated histones that do not alter the DNA sequence and determine whether genes are expressed or silenced. They add to the information content of the DNA by contributing to cell type specific patterns of gene expression. I like to think of the epigenetics as the genome's highlighter. In essence, epigenetic marks highlight certain genes to be expressed or silenced appropriately so that our one genome can be utilized differently at various times in development and in various tissues. In fact, so some genes may be expressed, the green genes may only be expressed in brain, red might only be expressed in heart, blue might only be expressed in bone. Additional characteristics of these marks are that they are malleable and affected by the environment and disrupted in many disease states. One such group of disorders are the Mendelian disorders of the epigenetic machinery, also called chromatinopathies. This is a rapidly emerging group resulting from germline mutations in genes encoding components of the epigenetic machinery. The 70 genes responsible for these conditions in 2019 are shown in the wheel and are divided into categories based on whether they encode enzymatic writers that place epigenetic or chromatin marks, enzymatic erasers that remove these marks, non-enzymatic readers that interpret the marks, or chromatin remodeling enzymes which actively alter chromatin structure. In many cases, the enzymatic writers, erasers, and remodelers also have reader domains, and those that do are shown in gray and have been referred to as dual function components. This group of genes is highly dosage sensitive. The PLI score, or the probability of being loss of function intolerant score, ranges from 0 to 1, and anything greater than 0.9 is highly intolerant to loss of function variation. When all genes in the group are considered, 80% have a PLI score of greater than 0.9. And when just intellectual disability genes are considered, the PLI score is greater than 0.9 in about 90% of cases. The vast majority of conditions are inherited in an autosomal dominant manner, indicated in the central circle by an A for autosomal and the filled black circles around the periphery for dominant inheritance, though there are exceptions. Most result from de novo mutations with a mechanism of haploinsufficiency. The most common phenotypic features of these disorders are intellectual disability and growth abnormalities. Intellectual disability is indicated by the blue shading around the periphery of the wheel, and growth abnormalities are indicated by the orange shading. These two features co-occur in about 87% of cases. And in addition to the most common features, there are many additional tissue diverse manifestations, including findings like hypotonia, anxiety, ADHD, autism spectrum disorder, as well as musculoskeletal findings like scoliosis, craniofacial dysmorphisms, eye findings, immune dysfunction, and structural anomalies of various organs. Mendelian disorders of the epigenetic machinery are really genetic disorders with genome-wide epigenetic consequences. In the normal state, various components of the epigenetic machinery work together to allow some genes to be expressed, like gene 1, and others to be silenced, like gene 2, in any given cell type. In the normal situation, all components of the epigenetic machinery are functioning properly. So genes like gene 1 have writers of activating marks, erasers of silencing marks, readers of these marks, and chromatin remodelers. And the net effect is an open chromatin state and gene expression. At genes like gene 2, there are distinct components of the epigenetic machinery, including writers of silencing marks, erasers of activating marks, and readers of these marks, and the net effect is a more closed chromatin state and gene silencing. In the setting of a Mendelian disorder of the epigenetic machinery, the direct effect, if, for example, one particular component of the epigenetic machinery is not functioning properly, for example, the writer of an activating mark, if it cannot place its mark and recruit the other epigenetic machinery components, then an aberrant set of components is present, and the abnormal epigenetic marks are placed, and the gene is abnormally silenced. There are indirect effects of this as well, in that gene 2, which is normally silenced, becomes abnormally expressed because the epigenetic machinery components that should be turning that gene off are mislocalized to gene 1. And this prevents them from placing the silencing marks and generating the appropriate chromatin structure and silencing that gene. And this happens at many genes throughout the genome, causing broad epigenomic changes, including genome-wide DNA methylation changes that Beckham just spoke about. Now I would like to tell you about how we recently elucidated the first neurodevelopmental disorder of the DNA demethylation machinery, initially called TET3 deficiency, and now also referred to as Beck-Fahrner syndrome. DNA methylation, or 5-methylcytosine, is the quintessential epigenetic mark. Classically, it occurs at cytosines followed by guanines or CpG dinucleotides. The writers of this mark are the DNA methyltransferases. And the TET enzymes are the erasers of 5-methylcytosine that catalyze the first steps of active DNA demethylation. The TET family of dioxygenases oxidizes 5-methylcytosine, first to 5-hydroxymethylcytosine, and then through a series of additional intermediates, which are then removed and replaced by unmethylated cytosine, completing the active DNA demethylation process. We elucidated TET3 deficiency or Beck-Fahrner syndrome using exome sequencing and gene matcher. We identified 11 affected individuals from 8 families with variants in TET3, which is important in embryologic development and neuronal functioning. Affected individuals from families 1 through 3 had biallelic variants inherited in trans from carrier parents, which we initially thought was consistent with autosomal recessive inheritance, though one or both parents in each family had milder manifestations. Affected individuals from families 4 through 8 had monoallelic variants, and all monoallelic variants occurred de novo with the exception of family 7, in which the proband inherited the variant from a similarly affected father. Affected individuals had highly similar phenotypes, which were consistent with other Mendelian disorders of the epigenetic machinery, including developmental delay and/or intellectual disability, growth abnormalities, hypotonia, autism spectrum disorder or autistic features, movement disorders, and seizures and/or EEG abnormalities. They also had characteristic craniofacial features, including a tall and/or broad forehead, brachycephaly, and a long hypotonic face, in some cases protruding ears as well. TET3 variants identified in the affected individuals are novel or rare, and here the monoallelic variants are shown in purple and biallelic variants are shown in orange. The underlying variants occur in the last exon, and the italicized orange variant is outside the catalytic domain and no longer thought to be pathogenic. In collaboration with Roberto Benezio's group at the University of Pennsylvania, we performed in vitro functional studies to test the catalytic activity of a subset of the variants. Briefly, TET3 variants or controls were overexpressed in HEC2-9-3 cells, and the ability to convert 5-methylcytosine to 5-hydroxymethylcytosine, the first step in active DNA demethylation, was measured using an antibody that specifically recognizes 5-hydroxymethylcytosine on dot blot assays. 5-hydroxymethylcytosine levels were measured in cells expressing each of 5 TET3 variants, indicated by the asterisks, and compared to 5-hydroxymethylcytosine levels in cells expressing wild-type TET3 or a known catalytic dead control in the last lanes. Quantification revealed that all variants tested showed reduced catalytic activity, with the exception of one, and that was similar to the known catalytic dead control. And this one is the arginine 752 cysteine variant, which I mentioned on the previous slide, we no longer think is fully pathogenic. And these data show that most TET3 variants exhibit hypomorphic activity in vitro and suggest that the disease mechanism is due to reduced TET enzyme activity. Many Mendelian disorders of the epigenetic machinery have specific genome-wide DNA methylation profiles or episignatures in blood, which I believe you just heard about from Beckham-Sudokovic. And we wanted to determine if deficiency of TET3 is associated with a DNA methylation signature. And this is a slide I borrowed from one of Beckham's reviews, so I won't go through it because he may have already gone through it. But the idea is simply that healthy individuals have a particular reference DNA methylation signature, and individuals with different syndromes, depending on which component of the epigenetic machinery or other components are defective, have distinct episignatures. So we hypothesized that deficiency of the TET3 eraser of DNA methylation would lead to an increase in DNA methylation and have a characteristic DNA methylation profile. We performed genome-wide DNA methylation profiling of whole blood from a discovery cohort consisting of 3 individuals with biallelic TET3 variants, 3 individuals with monoallelic TET3 variants, and 5 age and sex-matched controls for each affected individual. We used Illumina's EPIC bead chip array, which contains over 850,000 probes, and subsequent analyses were performed in Beckham-Sudokovic's lab. Hierarchical clustering analysis is shown, and each row represents an individual CPG site, and each column represents an individual patient sample. And the genotypes are color-coded based on what's shown here. Individuals with biallelic TET3 variants are shown in blue, individuals with monoallelic TET3 variants are shown in red, and age and sex match controls are shown in green. The DNA methylation level or beta value, which is shown ranging from dark blue to dark red, is shown here with this color scale. And 0 or dark blue indicates no methylation, and 1 or dark red indicates fully methylated. The hierarchical clustering reveals an overall increase in DNA methylation and clearly separates the TET3 samples from controls, and even separates the biallelic TET3 samples from the monoallelic samples. And the biallelic samples tend to show more of an increase in DNA methylation. Multidimensional scaling, like principal components analysis, groups the samples based on similarity. This clustered the samples into 3 groups, with biallelic samples shown in blue clustered on the far left, controls shown in green clustered on the far right, and monoallelic samples in red clustered in between. These data established a DNA methylation signature for TET3 deficiency, which separates TET3 deficient samples from controls and can further distinguish individuals with monoallelic and biallelic variants. To validate the TET3 episignature, we used a distinct cohort consisting of individuals with known monoallelic pathogenic TET3 variants or unaffected family member controls known not to have the familial TET3 variant, and the benign missense variant that did not reduce catalytic activity in our in vitro assay. Here, the original discovery cohort samples are shown in faded colors, while the new validation samples are shown in bright colors. Multidimensional scaling reveals that all the samples validated with the new samples with monoallelic pathogenic variants clustering with prior samples with monoallelic pathogenic variants, shown in shades of red, the new family member controls clustering with the original age and sex matched controls, shown in green, and the benign variant that didn't reduce catalytic activity clustering in between, shown in yellow. And we use these data to train a support vector machine to generate methylation variant pathogenicity prediction scores, or MVP scores. And these are the probability that a particular variant has the TET3 episignature, or the probability that a particular variant is pathogenic. The biallelic samples had scores near 1, and controls had scores near 0. The monoallelic pathogenic samples had scores between 0.5 and 0.8. And the benign variant was, again, in between. We then refined the TET3 episignature using both the discovery and validation cohort samples and their match controls, again here shown in faded colors. We used the new signature to test a new cohort, a testing cohort, consisting of biallelic and monoallelic TET3 variants of uncertain significance, shown in orange and purple. Both biallelic samples shown in orange and the 4 monoallelic samples labeled 19, 20, 23, and 24 shown in purple clustered with controls shown in green, suggesting non-pathogenicity. The other 4 individuals with monoallelic variants of uncertain significance, labeled 21, 22, 25, and 26, clustered with monoallelic pathogenic samples shown in light red, indicating pathogenicity. And the TET3 episignature was even able to identify the sample shown in black, in which was actually not known to have a TET3 DNA sequence variant. The episignature in this case was used to screen a database of over 1,000 undiagnosed individuals and identified this individual. When we went back, that individual's whole exome sequencing showed a nonsense variant in TET3. So this illustrates the robustness and diagnostic utility of the TET3 episignature. The methylation variant pathogenicity scores supported the other analyses. And when compared to other more traditional metrics used to classify variants, our DNA methylation signature correctly categorized all variants of uncertain significance. If we compare it to presence or absence in NOMAD, the CAD score, whether it's within or outside the catalytic domain, and the inheritance and segregation within the families. Taking a one against all approach, we next compared the TET3 samples to samples from other neurodevelopmental disorders with distinct episignatures and controls to determine whether the TET3 deficient episignature would be able to distinguish Beck-Fahrner syndrome from other conditions with known episignatures. 75% of the samples were used for training, shown in blue, and 25% were used for testing, shown in gray. And we found that the samples from controls and all other disorders, with the exception of one, had scores near 0. This indicates the TET3 episignature was able to differentiate individuals with pathogenic variants in TET3 from individuals with other neurodevelopmental disorders, with one exception. And the exception is the disorder called ADCADN, or autosomal dominant cerebellar ataxia, deafness, and narcolepsy syndrome, which is caused by hypermorphic mutations in DNMT1, a writer of DNA methylation. MVP scores for ADCADN partially overlap those with Beck-Fahrner syndrome. So we directly compared methylation changes in samples with TET3 and DNMT1 variants and their respective controls. Interestingly, hypomorphic mutations in the TET3 eraser of DNA methylation in Beck-Fahrner syndrome and hypermorphic mutations in the DNMT1 writer of DNA methylation in ADCADN would both be expected to cause an overall increase in DNA methylation, which is what we observed. Compared to controls shown in green, samples with pathogenic TET3 variants shown in red, blue, and purple, and ADCADN samples shown in black have an overall increase in DNA methylation. And the multidimensional scaling agrees and shows that ADCADN and Beck-Fahrner syndrome samples cluster separately, and that they both cluster separately from controls, indicating unique episignatures that can distinguish individuals with the two disorders. We now know of 28 individuals with Beck-Fahrner syndrome that have been described in the literature, and we actually know of many more, which we are currently writing up, and we have additional ongoing research to elucidate the neurodevelopmental phenotype in affected individuals and to use patient samples to study disease mechanisms and test therapies. So if anyone is aware of patients with this disorder, please send them our way. Now I want to share a bit of data from our multidisciplinary epigenetics and chromatin clinic at Johns Hopkins, which was founded in 2012 by Hans Bjornson, and with me as the current director. We have 2 affiliated genetic counselors, Jacqueline Britton and Carolyn Applegate, and the clinic became multidisciplinary in 2018 when Dr. Jacqueline Harris, a neurologist and neurodevelopmental specialist, joined our team. Currently, anyone who comes to our clinic sees me as the clinical geneticist, Dr. Harris as the neurologist and neurodevelopmental specialist, and Jackie Britton as our genetic counselor. We completed 741 epigenetics and chromatin clinic visits in 5 years. And here, red indicates new patients, whereas blue indicates follow-up visits, with diagonal lines indicating video visits. Earlier on, a larger proportion of patients were new, but for the past 3 years, we've had a larger proportion of follow-ups, with 63% being follow-ups in year 5, suggesting that our patients are satisfied and appreciate the ongoing management. We saw 432 individual patients in 5 years, and the map shows the distribution in the US, with about 27% residing outside the state of Maryland, with over half of states being represented and 3 continents as well. 153 patients have epigenetic disorders, with 75% having a Mendelian disorder of the epigenetic machinery, or 115 having one of these conditions. 23.5% have a confirmed imprinting disorder, and only 27.5% remain undiagnosed. Of the 115 Mendelian disorders of the epigenetic machinery we have seen, the top diagnoses are Kabuki syndrome type 1, Wiedemann-Steiner syndrome, Soto syndrome, Arboleda-Than syndrome, and intellectual developmental disorder autosomal dominant 1, or MBD5 associated neurodevelopmental disorder is another name for that. The 115 patients with Mendelian disorders of the epigenetic machinery have mutations in 26 distinct epigenetic machinery genes, with the highest proportion involving writers of these marks, then erasers shown in blue, readers shown in yellow, and remodelers shown in orange, with the dotted sections indicating the proportion of dual function components. So we do see a wide variety of patients in this group. In these 115 individuals with Mendelian disorders of the epigenetic machinery, we see a variety of neurodevelopmental phenotypes, which are consistent with those reported in the literature. The bars indicate the total number of affected individuals, and the individual colors indicate diagnoses, with the top 5 being shown separately and the others being lumped together into the other category shown in aqua. The most common diagnosis is cognitive impairment, which we saw in 100% of individuals with Mendelian disorders of the epigenetic machinery. And most had mild ID, but there was a range from developmental delay with not quite meeting intellectual disability to severe to profound intellectual disability. The second most common neurodevelopmental diagnosis was hypotonia, followed by ADHD and anxiety, and seizures and autism spectrum disorder were slightly less common. Finally, about 70% of individuals with these disorders from our clinic have a growth abnormality. About 78% have growth retardation, and about 22% have overgrowth, with growth retardation more often affecting height compared to head circumference, and overgrowth impacting them similarly and often impacting both. So in summary, the Mendelian disorders of the epigenetic machinery are a rapidly growing group of disorders due to germline mutations in epigenetic machinery and chromatin-modifying genes. Most are autosomal dominant due to haploinsufficiency, and the genes are highly dosage sensitive. Most common phenotypic features are intellectual disability and growth abnormalities, but many tissue-diverse manifestations are identified. TET3 deficiency or Beck-Fahrner syndrome is a new disorder in the group implicating the DNA demethylation eraser system, and it includes behavioral differences, epilepsy, facial features, autism, hypotonia, retardation of psychomotor development, and size differences. It has a sensitive and specific genome-wide DNA hypermethylation profile that's important diagnostically. And many of our observations have come from our multidisciplinary epigenetics and chromatin clinic at Johns Hopkins. So thank you all for your attention. I'd like to thank my collaborators, my lab, the Epigenetics and Chromatin Clinic team, our funding sources, and most of all, the patients and their families. Thank you so much.